But what was important is then at the end of this particular initial investigation, we had a very novel microbial culture. It was actually a consortium. We had one particular component which was a rod shape, which was Clostridium 1, and another component was a cocci shape, and this particular cocci shaped was the Enterococcus species that we were able to get from this particular consortium. And then we had this thermostable nutrient suit. And if you see this, after incubating it at 94 degrees Celsius for up to 21 days, it still was not getting denatured. You would still see the clear nature of this particular nutrient medium, and you will see the growth that this particular medium has supported, and it has produced all these things. Once you know that this particular culture is able to produce this, you should confirm its ability to extract oil. And for that particular purpose, we did some sandpack studies. I will show you some more pictures of this, and we did the core flood studies. Now, what is the significance of sandpack and core flood studies is, this is where you can raise the temperature, you can raise the pressure. So temperature can be taken very easily up to 95 or 100 degrees Celsius in this particular one, and you can also increase the pressure, because in laboratory it is not very easy to increase the pressure and ensure the growth. And in this particular case, what we were able to do was take the pressure up to 2000 psi and demonstrate its ability to grow and produce metabolites and mobilize the residual oil that was remaining there. So in this particular picture, you would see this is the colony of the organism, and you will see these sandpack studies. And these were the columns. This was the control and this was the test. And you will see that in this control column, oil was not recovered. Only medium was put into this. And here, along with medium, we put our culture, and it mobilized the oil. If I show you this particular picture, you will be able to appreciate that this is a test column, and you will see that only sand is remaining. And most of the oil from this sand was mobilized, while in this control you will see that oil part was still remaining there. It actually highlights the ability of the culture to not only grow there, produce metabolite, and mobilize the residual oil. Now, once it is done, then you have to go ahead and you have to demonstrate applicability of the culture in field. So for that particular purpose, we decided to carry out some of the pilot scales studies. And for pilot scale, we had some discussion with ONGC, and I had certain numbers in my mind. So the ONGC people asked me that if you have to carry out pilot studies, what is the scale that you have in mind? And I said 100. I was just little hesitant, because doing any trial at 100 liter would also take some doing. And those people said, okay, 100,000 is fine with us. And I said, what's 100,000? And they said, 100,000 liters. It's a small trial, but okay, we'll do with this. And the question started coming to my mind, 100,000 liter, preparing the medium, preparing the inoculum for that, how are we going to do that? And that actually made me realize the challenges that one face in applying any technology or microbial process in the field. So I wanted to share some of those pictures with you. So I talked to the ONGC people, I talked to some of the local vendors, and somehow we were able to manage all these processes, and this is what we would show. That for preparing this, ultimately we decided to conduct these trials at 100,000 liter capacity. So for that purpose, we decided we had to get the water. And these are the tankers from which we could get. This is a full tanker, which has only 16,000 capacity. So we had to get 9 such tankers, from which we could get the required volumes of the water. Once we had, then another part is how are you going to mix the chemicals? So for that purpose, we got something which is similar to this. Usually it is used for preparing the concrete mixture. So we got that. It has a mixer inside, and in that we were just putting those chemicals in bulk. And then the whole thing was, after it was mixed here, it was transferred. Before that, we had to sterilize it. And for sterilization, we used this particular assembly. This particular assembly had the filters in that. So we would put all the ingredients in this tanker, and with the help of pump, we would push it through this. And from that, the whole thing would get sterilized. And after getting sterilized, we had to store this. So these were the tankers in which we stored the sterile, so-called sterile medium. And these were the cultures which we had grown in the laboratory, up to 700 liters of the cultures. These cultures we mixed into this, and after that, the whole thing was then injected in the oil well. Actually, all other people did the task, whatever was there, and I was just standing there to take the credit for it. Whatever important was this, that after it was injected, then we should close that oil well for some time. And after closing the oil well for 14 days, after that we opened it. And when we opened it, only gas came out of it. And that gas came out for like half an hour. It was just making that whoosh kind of noise, and the entire gas was coming out. And after that, we started using the pump, and after that, we started recovering the oil. So this was the dead oil well. Only water was coming out of it. And after that, after 14 days of incubation, we started getting oil. And that oil was 1 or 2 meter cubes per day, and it went on for more than 90 days. So for the figures of first 90 days were the ones that we have taken into consideration, that whether this really was a very effective or economical job or not. So for that purpose, we did some economic analysis. So for the nutrient medium, now this was the nutrient medium which we used, and my naivety again spoke there, because whatever chemicals we had to procure, we procured chemicals. I was used to it, so I wrote everything like NLR grade reagents, and it really costed me 3 lakhs of rupees for this nutrient suit. And now today I know that I can get the bulk chemicals in the commercial one, and the same medium now cost me only 22,000 rupees. So that is the difference between the laboratory grade reagent price and this. And then the infrastructure, it really cost us, because getting all those waters and all those pumps and filters and everything has its own cost. So total cost was 6 lakhs rupees. And then after that, within 90 days, we recovered 899 barrels of oil. And at that time, it was actually done in 2014. And if we consider that, we still were able to recover crude oil, and crude oil worth 63 lakhs of rupees. Now, if you imagine today's price, this profit margin goes more than double. And just imagine that 6 lakhs of rupees, within 90 days, you are recovering more than 63 lakhs rupees, more than 10 times return on the investment. And I could very safely assure you that there are very, very few such ventures where you could get such high returns over this. Now, what is interesting is once you have that, then the real problem comes. You go to investors and you say that, okay, we want this. And then they ask, so what exactly is your commodity? And I said, we are recovering oil. So what is your product? And I said, we have process. So I said, is it a process? Is it a crude oil? Is it the culture? Is it the nutrient medium? What is it that you are trying to sell? And as a microbiologist, you are at a loss, because you have developed a microbial process, and this microbial process is recovering oil. But when you talk to the financial people, their questions are totally different. So you have to tell them that my end product is actually crude oil. I will be selling you crude oil. And they say that, okay, the ONGC is selling crude oil. What is your difference? I say that I develop crude oil from the waste of the ONGC. There are these oil wells, ONGC is not handling anymore, and that is my target. From waste, I am generating wealth. And now they are interested, and they say, okay, this is something which is interesting, and people are ready to take this and take it further. But before we do that, we have to carry out SWOT analysis. What are the strengths of our technology? The finance guys are always going to ask us, and this is where we really lack, because we are the microbiologists, we are the scientists, and what we do is we develop the process, and we expect industry to take it as is and where is basis. And industry would never do that, and that is why we always see very low level of transition of technologies from the laboratory to field. And for that purpose, one needs to actually do some kind of analysis, and this is the analysis of strength, weaknesses, and we have to convince them. So what is the strength of our technology? It is a cutting edge technology, a kind of monopoly, because nobody has this culture which grows above 90 degrees and which affect oil recovery, and it is an economically attractive one. We have shown its demonstration, so we have the proof of concept. And we have already established and reputed end user in the form of ONGC who has used it and kind of certified it. And we have now established expertise. So these are our strength. But at the same time, there are certain weaknesses. And weaknesses are this, that if at all I have to take it to the field, I will initially need lot of capital investment. And who's going to do that? As a research institute, I will not be able to do that. And another important part is the reservoir characteristics, that you have this process, but you have to implement in the field. We had some very weird experiences in one of the oil wells where we put in our nutrient medium, we waited for 14 days and we opened and nothing happened. No gas, no oil, nothing. And then we had to do lot of analysis, and then we found out that that particular reservoir was a carbonaceous reservoir. And when these microorganisms grew and when they produce acid, that acid actually dissolved some of those carbonaceous rocks, and there was a channel which was created. And through channel, that entire medium got diverted to other part of the reservoir where there was no oil and there was no production. So now you have to take into consideration what are the reservoir properties, whether the type of the rock that is present there, the porosity. Sometimes you find out that porosity is very minimum. So it is like 10 to 15 microns. So microorganisms will go there, they will start multiplying, but they don't have enough space to multiply there. So their growth will be inhibited. And these are the problems which will not anticipate in the laboratory. For that purpose, you have to go to the field, implement it, and then you understand this. So these are some of the weaknesses, but there are humongous opportunities there. The market is growing. You have now the well-established recovery process, and there is an increasing demand for the exploration of oil wells. Now, if you see that demand for the oil every day is going up, and this is going to last for only maybe a couple of decades. Very soon we are experiencing a transition where everybody is shifting from the petroleum-based recovery method to petroleum-based fuel to say non-petroleum. For example, biomethane, bio-CNG could be an alternative one, or maybe the ethanol blended could be one, or solar could be there. And these actually are our threats. The political instability, even internationally, is a threat. Highly competitive market at lower temperature is a threat. Failing prices of crude oil is a threat. And you know, the oil prices are only increasing in India, not outside India. Outside India, they are still dropping. And in spite of that, we are still paying higher because of some other reasons. But the failing price of crude oil is another problem. And with the development of the solar energy, bio-CNG, and ethanol, that could be the threat. But now that we have the process and we have only maybe 20, 30 years to use this is the time for us to implement in the field. Now for doing that, one must understand what exactly is the market potential. And you will see that even this, the 6.3% of the GDP India's GDP was actually invested in getting the oil from this. 75% of the oil is imported by our country. And whatever the reserves are there, and even if we go by the extraction of 9 barrels per day, which was a very small quantity we have taken it, we would still extract 6,900 barrels. I mean, if this many oil wells are used for this particular one can extract these many barrels, and it could save so much of foreign exchange for our country. Now that means that it's a huge, huge potential there. So in that case, there is a huge technology. Now what is important for us to understand that we have one process. We have developed it. It can be implemented. What, for some reason, if this process doesn't work? The microbial culture that we have developed, what if it doesn't survive in some of the oil wells? So one must have a fallback strategy. And one should start thinking about that fallback strategy today only. So keeping that in mind, we also decided to explore some other kind of opportunity. Now, what was that opportunity, or what was that particular task was this, that we have to develop a culture. That culture has to survive and grow in the oil wells. Every oil well will have different set of temperature, pressure, and other chemical structure, everything. So those properties of oil reservoir will vary from one reservoir to another reservoir. Now, the microorganisms that will grow best will be the ones which are native to that flora, because they are already acclimatized, they are already adapted to those reservoirs. Why can't we use that? And if at all we have to use that, how can we do that? Now for that particular purpose, we have decided to use one particular approach. And we decided to now, after experiencing some success in that, we have already named it as Petrogenomics approach. Now what we do here is very simple, that we collect a water sample from that, and from that we want to use their indigenous microbial flora. So what are these indigenous microflora? They are better adapted. There is less energy consumption than the externally added MER, and lower cost, because you don't have to grow the culture, you just have to inject the nutrient medium into it and use the microorganisms which are native to the reservoir to grow. So for that particular purpose, we decided to use this. Collect a water sample, isolate metagenome from that, sequence the metagenome, analyze that metagenome. What type of microbes are present, whether they have the ability to produce desired metabolites, and based on that whether we can design the nutrient medium that would be suitable to that. For example, you have microbial culture which is a thermophilic, which has ability to produce all these metabolites and which has ability to produce even amylase enzyme. Now it really changes the total dynamics. What it does is, if it is producing amylase enzyme, I can use starch. If I use starch, it will be thermostable. It will be very cheap. If I use from starch industry the waste material also, that can be used as a nutrient medium. So using that, I can directly reduce the cost of the nutrient medium. I can reduce the cost of cultivation. I can reduce the cost of sterilization of the media. The whole thing I can mix and inject into the oil reservoir. So we wanted to study that, and this is just to show you that these are the 6 different oil wells from which we collected the water sample. Each of the oil well was characterized taxonomically as well as functionally distinct microbial population, and from that we did this analysis. Now what was interesting was that even though these all 6 samples were collected from Mumbai High region, only 2, that is F1 and F7, were the ones which showed the presence of the thermophilic microorganisms. All other ones showed the presence of mesophilic organisms. What it meant was that only these 2 oil wells were the ones where we could use the inherent flora or indigenous flora for the application. Now, if that was the case, we also did their functional characterization, and we were able to detect the metabolites like carbohydrate metabolism byproducts as well as EPS1. This is because this tells us their ability of the cultures to produce solvents, volatile fatty acids, and carbon dioxide. And you will see whatever genes were there in that metagenome, we trace them back to their origin, and only in first 2 ones we were able to trace them to the thermophiles. In other cases, it was traced to mesophilic ones. So what it told us was this, that only these 2 were the oil wells where one could use the metagenome approach, one could use the indigenous flora for exploration of oil well. Now, if this hypothesis was true, then if I use the nutrient medium and if I try to cultivate the organisms, I should be getting growth only from F1 and F7. And this is precisely what happened. Only from F1 and F7 we got the growth of the organisms, and we also got the production of the desired metabolites. While in others 4 oil wells there was no growth. That was a proof of concept of using the approach. And using this particular approach, now we are in a position to go for MUR by biostimulation approach. And we have already started a procedure for filing a patent on this particular one. And now we have named this particular one as a Petro Genomics approach for the production of the oil from the depleted oil reservoir using the indigenous flora. And this is in general the flowchart that I have explained it here. So this is a very nice way in which one could go on exploring these particular ones, the organisms from the oil reservoir for different approaches. Now quickly I will go through some of the other applications of these microorganisms, and it is very interesting one. In this particular one, we also detected presence of some of the thermophilic methanogens. And this was a very interesting one, that the methanogens were present there. For MUR, they were of no use. But then we just had another project. And in another project, what we have been doing is trying to generate energy, that is methane, from the rice straw. And if rice straw is a renewable one, it is abundant, it is very cheap. And if at all we use that rice straw as a fuel, then we can generate huge quantities of methane, and that methane can be used either for generation of electricity, or it can be used in the form of bio-CNG as a vehicular fuel. So that is precisely what we wanted to do. Now, in this particular case, the drawback or the bottleneck is the pre-treatment requirement, that if at all that methane, rice straw is to be used for biomethanation purpose, it has to be pre-treated at high temperature with alkali. And we don't want to do that because it is cost intensive, it is polluting one. So we developed a microbial consortium using anaerobic fungi, and with the help of that, we were able to develop the process. And this particular process was able to generate methane without any pre-treatment requirement. So it was able to do it at ambient temperature, and it was able to do it without any pre-treatment. But the important aspect what we realized was, Instead of 37 degrees Celsius, if we perform the same process at 55 degrees Celsius, then the hydrolysis of rice straw was much more efficient. And for that particular purpose, we needed thermophilic methanogens. So we decided to supplement these particular reactors with the thermophilic methanogens isolated from the oil reservoirs. And when we did that, we were able to get more than 334 liters per kg Vs of the biogas. Now you will ask me what the certain questions, that what is the significance of this particular number? This is more than 95% of the theoretical yield, and we are getting more than 95% of the theoretical yield of methane at this particular temperature without any thermochemical treatment. So you will ask me the question, what is the significance of that? And I will explain you only in 2 sentences. The cost involved in the pretreatment for 100 tons per day capacity plant, which we have been doing with Melham, it's an Indo-French company. So 100 tons per day capacity cost for the pretreatment comes to around 44 crore rupees per annum. The biogas that is generated from this reactor is 45 crores. So roughly you split equal. That means whatever money that you are putting it is the money that you are getting it. By using these thermophilic methanogens and by doing this approach, the cost of pretreatment and biomethanation, the entire process cost has come down to 4 crores. And the biomethanation cost has gone up to 55 crores. So now from 4 crores investment, we are getting 55 crore rupees of methane. That is 51 crore rupees per annum of benefit only through this. And this is the cost of methane. You get organic fertilizer because the slurry that is developed from that can be recycled as an organic fertilizer. That benefit is even extra. Now this is totally a different approach that we got application. So that's why I call it a cool application of these hot organisms which were totally isolated from this. And there were certain other applications, and I will not go into details of that, but only one last application that I would like to share with you and stop there. And that is this particular one. And this is a very interesting one that we found out, that many of the oil reservoirs, they face the problem of reservoir soaring. Now what is reservoir soaring is, they have the sulfate reducing bacteria. Many times whatever water is injected into that, it contains sulfate. So sulfate reducing bacteria will convert that sulfate into hydrogen sulfide. And hydrogen sulfide being highly corrosive one, it will corrode the pipes and it will really cause a lot of problems. Hydrogen sulfide is also health hazard, and people working with this will also be getting adversely affected. Their health will also be inversely affected. Now just to give you how serious is the problem, the budget of the oil industry for control of sulfate reducing bacteria or reservoir soaring per year is $2.3 billion. They are spending so much of money on control of that, and still they are not able to solve this problem because bacteria keep on developing resistance to that. Now what we did was, from the same samples, we isolated a bacteriophage, and this bacteriophage is able to infect the sulfate reducing bacteria. We all know bacteriophages. These are the viruses which infect those bacteria. They cause the lysis of that. So what they are doing is, they will inject, they will go inside, they will inject their nucleic acids into sulfate reducing bacteria, which will multiply, synthesize thousands of such particles, virus particles. It will burst the cell. They will come out and they will start infecting others. And what we were able to isolate such bacteriophage, and this bacteriophage was very specific in its host range, meaning that it was able to lyse several species of sulfate reducing bacteria, but other bacteria which are unrelated, it did not infect any of those bacteria, indicating this one. And you will see that this is what the blackening happens because of the H2S production. And when we add these bacteriophages, you will see the clear one. So what essentially we were able to achieve was more than 90% of the reduction or inhibition of sulfate reducing bacteria within 24 hours of inoculation of this. And that effect lasted for 28 days. You use chemicals, the effect lasts for only 48 hours. So every alternate days you have to go on injecting this. With this, we can have only. And the price of this is absolutely nothing, because this is cultivated only using seawater and sulfate reducing bacteria. And this is one technology that we are now implementing in Bombay High Reservoir. Again, isolated from the reservoirs and used for the benefit of that. So there are other bioremediation approaches, but I will not go into details of that in front of Dr. Deka. He can take care of that. But this is the one that we have done so far using these microorganisms. So right now, these microorganisms that we have developed, we call them as two different processes. Petro B is the microbial culture and the nutrient medium that we have developed, and Petrogenomics is our approach. And this is our kind of vision that ARI will be the head office of this Petro B, where you can come and get a cheap oil. So thank you very much for giving me this opportunity to share some of our research. And I would like to wish the bestest of best luck to Dr. Deka on his retired but still very active research life, even beyond his superannuation. Thank you very much.